Okay, let's see. Yeah, thank you so much for the introduction. Thank you for the invitation to be here today. I really appreciate it. And the title of my talk today is, sorry, I have a laptop here and then my big screen is here. So I'm mostly looking over here. Anyway, the title of the talk is Hadal Microbial Ecology, Biogeochemistry, and Microbial Enzymes at High Pressures. I do a range of different things, but what I will highlight in this specific talk actually is one of the things that I do here at the Hadal Center. So the center is dedicated to studying the deepest parts of the ocean, which is Hadal environments, which is what I'll be introducing as well in this talk. I would like to begin with a slide on learning objectives and our proposed agenda for this talk. And in terms of the learning objectives, I hope that we take away from this talk the ability to understand the distribution of Hadal environments and their unique bathymetry, characterize or identify the factors that influence life and the processes within Hadal environments, identify some of the defining features of Hadal microbial ecology and biogeochemistry, and learn about some ongoing projects related to high pressure in Hadal environments. And so our roadmap or our agenda for this talk is an introduction of Hadal environments. It's going to be fairly jam-packed, trying to answer the questions of where are these environments, how are they defined, and what's so unique about them. Asking the question of and answering, hopefully, what's known about Hadal biogeochemistry. And the focus that I will have in this talk is actually looking at the carbon cycle and how these Hadal environments are considered as depot centers or deposit centers of organic carbon, and how that could lead to very high oxygen / carbon demand by microbial communities, and how these areas are considered hotspots of activity as a result. And also we'll look into what's known about Hadal microbiology from surface to Hadal bottom water. So we'll take a look at the pelagic microbial communities, something that is very near and dear in my heart. I mostly study the water column, but nowadays I'm studying more of the sediment. So I'm also going to be introducing some work as well from surface to subsurface sediment. And then that'll be the primer that leads to the current research that I'll talk about, which is the pressure effects on microbial enzymes that break down organic matter. So we're going to marry the two, microbial ecology and biogeochemistry. So there's been actually quite a bit of interest in Hadal environments recently, in large part because of increasing accessibility to these environments due to new technologies. So Alan Jamieson put out this perspectives paper that looks at how many papers have been published from a few decades ago all the way to about 2017. And you can see that it's seeing this exponential increase. And in part, it's driven by things like submersibles that can go down to deep waters. And this limiting factor, for example, has a depth capacity or depth range of all the way down to 11,000 meters, so 11 kilometers, which is the deepest part of the ocean. And as well, there are these autonomous vehicles. One of them is a benthic lander that can be deployed, be separated from a line right here, and it can take various measurements. And I'll be presenting some work that's been carried out using benthic landers, as well as multi-core. And this is something that's attached to the winch of the ship, but it brings back these gorgeous sediment cores all the way down to also 10,000 meters of sediment. So what are Hadal environments? Essentially, they're environments in the ocean that span 6 to 11 kilometers deep. So when we're thinking about the water column, right? So in the upper layer, in the photic zone, we're in the epipelagic. Then we transition into the mesopelagic, down to the bathypelagic until about 4,000 meters. And this is where the average oceanic depth is. Sorry, beneath that is abyssopelagic, so that's 4 kilometers down to 6 kilometers. And once we hit 6 kilometers, we begin the Hadal zone. And This is just a cross-section of the seafloor in Hadal environments. So going from 6,000 meters down to 11,000 meters, so the hadopelagic. And the sediment essentially, the seafloor essentially looks like a funnel. And it's because of these very steep slopes. And this, we will learn, is really important for concentrating material down. So similar to a sediment trap, for example, and it's designed that way in part to capture and to concentrate particles. The same, in effect, is what happens in Hadal environments. And then these Hadal environments are formed when two continental plates collide and one of them subducts underneath the other one. So in these subduction zones, you see these fairly deep trenches, right? And we can get down all the way to 11,000 meters. And these are the Hadal trenches that we're going to be learning about in this talk. And because there's, this is very high, because we're in subduction zones, it also means that there's high tectonic activity. And we'll learn today as well that high tectonic activity is one of the defining features of these Hadal trenches, and part of the reason why there's so much elevated carbon as well as elevated microbial community, despite what we would think that these are relatively barren places. They're really not. In terms of distribution of Hadal trenches on Earth, a lot of them are located around the Pacific Rim, right? So, but there are some as well in other parts of the world. So for example, the Aleutian Trench is, the South Sandwich Trench is in the Southern Ocean. So that's a little bit right here. I believe that's N. But most of the deepest ones actually are over here in the Pacific, and I've underlined which ones they are. The most familiar one that maybe we know of is the Marianas Trench, almost 11,000 meters, followed by the Tonga Trench, which is kind of north of New Zealand. And this one is near and dear my heart because I'm Filipino. The Philippine Trench is 10,540 meters, so on and so forth, and then followed by the Kuril-Kamchatka Trench as well as the Kermadec Trench. In terms of coverage of seafloor, Hadal environments are actually fairly minimally represented. So this is simply a distribution of the percent area that these different places, the coastal environments, continental slope, bathyal sediments, abyssal sediments, cover along the seafloor. So we know that the deepest part, on average, the ocean is about 4,000 meters. So that's the reason why we've got fairly high bars here in the bathyal and in abyssal. And then you can see that Hadal environments simply represent about 2.5% of the global ocean seafloor, which kind of raises the question of, well, why should we study Hadal if they represent such a minimal space in the ocean seafloor? And a lot has to do actually with the fact that, as I said previously, is that it's depot centers of carbon and it hosts high microbial activity. Here are some things that actually influence the life of organisms and the processes that occur in Hadal trenches. So things like geological age of these trenches, surface primary productivity, right? Global hydrodynamics, so the currents in these areas, proximity to landmass, that's very important, and we'll get to that in a little bit, as well as seismic activity, that's fairly coupled to proximity to landmass, as well as its topography. And one thing that I'd really like to highlight as well is that we've got fairly high hydrostatic pressures going all the way from atmospheric pressure, from the atmosphere and the surface area, surface waters, all the way down to 10,000 meters. So that's also one of the defining features of Hadal trenches is its very high hydrostatic pressure. One thing I'd like to focus on in terms of the factors that influence life in Hadal trenches is surface primary productivity. And essentially, we may be fairly familiar with this, but just in case, the biological carbon pump, which is that process that takes carbon dioxide from the atmosphere, fixed into organic carbon by primary producers in surface waters, and then exports that down to the deep water column, is the process that links surface water to Hadal environments. And there's a lot of ongoing processes there, some of which I also have quite a bit of research interest in. But essentially, this process can bring and export out of the upper water column quite a bit of carbon, which means that there's possibly quite a bit of carbon that can make its way all the way down to the deep sea, as well as Hadal trenches. And just to kind of see how much carbon 6 to 10 petagrams of carbon per year may look like, here is a very dense marine snow in Sagami Bay. This is off the coast of Japan at about 1,500 meters. And you can see all of these are marine snow. So in highly productive waters, particularly in coastal waters, we can have very high amounts of marine snow being exported. The next thing that I wanted to focus on, kind of leaving the water column and into sediments, is this idea that Hadal sediments are depot centers of carbon. And what we mean by that is that there's a lot of carbon that reaches Hadal sediments, much more so than what we would anticipate based on its depth. And it's related to the fact that it's shaped like a funnel. And essentially, a lot of things makes its way down to the deepest parts of the ocean. And so this study by Kazumasa Ogori that just came out looks at the percent burial efficiency of organic carbon, as well as the MAR, or mass accumulation rate. And they did this in 3 different trenches. So in the Atacama Trench, the Kermadec Trench, and the Mariana Trench. And what they did was they compared the values that they found to values that's been published in literature by Don Canfield, as well as Ronnie Gloon, both at SDU. And these data that they've previously published comes from 10 to 5,330 meters. So that's also, it has the percent burial efficiency of organic carbon, as well as the mass accumulation rate, or how quickly carbon can accumulate in sediments. And what we find, or what they find, sorry, is that trenches are all the way at the higher end of this trend. So they have very high burial efficiency of organic carbon, and they have also, as a result, high mass accumulation rate. So there's a lot of carbon relative to other parts of the ocean seafloor. There's a lot of carbon that's reaching the sediments in Hadal trenches. And one of the underlying mechanisms that delivers carbon to these Hadal sediments is actually mass wasting events. And mass wasting events essentially are anything from landslides or earthquakes that kind of displace items and then deliver these items to another place. That's essentially what a mass wasting event is. And what Kazumasa Ogori back in 2013 did, the research that he carried out was looking at the Japan Trench. And there was an earthquake in Japan in 2011. And they set out to deploy some videos, some cameras, and we'll look at a video in the next slide. And they also took some sediment core samples from the Japan Trench at around 7,500 meters. And they did some X-ray CT scan here. And just to read this, the CT value increases with density of the sediment that's deposited in that layer. And they also measured lead-210 profiles. And for those of us maybe not familiar with this, lead-210 profiles indicate the sedimentation rate of in sediments, of sinking organic matter, because lead-210 only comes from the atmosphere. Therefore, it's delivered to sediments by sedimentation of particles, such that when you've got high lead-210 concentrations, that indicates very recent deposition. So they look at the sediment core and the lead-210 profiles within, and they notice 2 different layers. One is uniform values, which indicates fairly rapid and homogeneous deposition. That's because of this earthquake. It sort of moved a lot of things from shallower areas and into the deep parts of the Japan Trench. And then this other layer that shows this gradual decrease, which is very indicative of gradual deposition, right? So you've got fairly newly deposited sediments up here. And as you go down in the sediment, then lead-210 values decrease, indicating older and older sediments. So this is what you would expect with more gradual sediment deposition that's characteristic of much of the world's oceans. But when you've got mass wasting events, this is a profile that you see. And recently, Kazumasa is killing it. Kazumasa is also did this study, this calculation, and showed that mass wasting events can increase carbon accumulation by a factor of 4 because it delivers so much of that carbon that's stored in sediments from shallower areas to deeper areas, which is just so fascinating. Here's a video of Hadal, two Hadal sites where they deploy these cameras to look at what's going on in these sites. I don't know if I can get this to play. I hope I can. Maybe I have to go back to my clicker. There you go. And what I want to move to is actually one of the sites which begins right here. Okay. What we are looking at is quite a lot of organic matter and sediment particles in the water column. This is fair. I mean, we are at 7,500 meters, and this is fairly atypical. You see an organism there as well. And this is essentially a nepheloid layer. So bottom water currents bringing quite a bit of organic matter, particles, and it's causing this turbidity. Very uncharacteristic of the deep sea, but they argue that this was captured on video because the earthquake displaced a lot of the sediment into deeper waters. So that's really nice evidence of mass wasting events influencing Hadal trenches. Now, what are the consequences of elevated deposition in Hadal sediments? And here we're going to get into a little bit of the biogeochemistry and then the microbial activity. This is a study conducted in the Mariana Trench, so the deepest part of the ocean in the Challenger Deep. And what they did was Ronnie Glued deployed these autonomous benthic landers that can measure oxygen concentrations in situ. And they did this at a reference site, so at an abyssal plain around 6 kilometers or 6,000 meters. And this is a fairly typical oxygen profile. So x-axis is oxygen concentration in micromolar, and then this is the interface, the seawater and sediment interface. So here they're bringing these sensors into the sediment. And what you can see is there's a gradual decrease of oxygen, indicating some level of microbial activity. Then they compared that to the sediments at Challenger Deep, which is the deepest part of the Mariana Trench at 10.9 kilometers. And you can see this whopping decrease of oxygen concentration from the surface of the sediment all the way down to about 16 centimeters. So this is very indicative of very high activities of microorganisms, right? So heterotrophic oxygen respiration uses up oxygen and is the reason why oxygen is drawn down. And this is so that they can utilize the existing carbon that's present in these environments. And they did a nice table, they made this nice table that compares several parameters, several sediment parameters between the reference site as well as a Challenger Deep, comparing, for example, oxygen uptake, so 85 micromoles per square meter per day in the reference site, 154 in the Challenger Deep, organic carbon content, 887 versus 1,108, chlorophyll A, phyton coming both from primary production, 136.20 versus 560, 1680, average prokaryotic density 0.14 versus 0.97. There is a lot of microorganisms in the very deep parts of the ocean. And then here's that sedimentation rate based on the excess 210, lead-210, 17,880 versus 5459. So this was published back in 2013, and still to this day, we continue to cite it because it's just such a striking comparison of how much activity there is in the deepest parts of the ocean compared to abyssal sites. He did a follow-up study where he measured activities using these benthic landers at multiple sites within a trench. And even within the trench, there's quite a bit of variability, but overall microbes in hadal sediments show about 2 to 6 times higher activities than adjacent abyssal sites. So then we move from, I keep seeing some questions in the chat. We move from biogeochemistry to the structure. And here, we're looking at a paper by Nuno Ora back in 2015. It was a PNAS paper where they looked at 16S sequences of microbial communities, all the way from surface waters down to 10,000 meters or a little bit deeper than that in the Mariana Trench. And essentially, we see this transition, or they see this transition from the mesopelagic and abyssopelagic into the hadal pelagic. And just to be a little bit more quantitative here, they provided these nice pairwise comparisons of microbial community similarity based on Bray-Curtis or dissimilarity, such that if it's one or white, there is no similarity. So there's very large distance between microbial communities. So when they compare, for example, the photic zone microbial communities to, the photic, how do I do this? The hadal, let's do this. The hadal to the photic, there's hardly any similarity, so the distance is large. And then also when you compare hadal sediment, hadal microbial communities to the mesopelagic, there is a little bit more similarity, hence you're getting some more of that color. So essentially it's saying that there's quite a bit of turnover as we go from shallower water column to the deeper parts of the water column. But what's interesting, and it's something that they didn't quite discuss in the paper, is based on UNIFRAC, which looks at presence absence as well as phylogenetic distances. Here in this analysis, you have to incorporate a phylogenetic tree. What they actually find is that hadal microbial communities, when you start to consider phylogenetic distances, actually look similar, show high similarity with photic microbial communities. Essentially, they're not the same organism, but they are fairly closely related. And just kind of qualitatively and visually, you can see that there's increased representation of gamma proteobacteria, which I believe is in the turquoise here, and Bacteroidetes, which is in the red in hadal environments, whereas you're those are fairly minimal representation in the meso to abyssopelagic, but then they sort of make a comeback. They're not apparently the same ones as Bray-Curtis dissimilarity index indicates, but they are very closely phylogenetically related. Then we move to microbial community structure in surface to subsurface hadal sediments. And I kind of want to begin by setting the stage here, looking at the redox conditions by measuring, by showing the oxygen concentrations, the nitrate, and the ammonia profiles. This is a paper by Clemens Schauberger, also here at University of Southern Denmark. And first, I show the abyssal sediment course. So we have a comparison here, and you can see that nitrate increases, let's start with oxygen. Oxygen decreases, nitrogen increases, and then there's eventually a gradual increase of ammonia. And this is simply their sectioning scheme of the sediment cores. So they take fairly high resolution in the upper 10 centimeters, and then from there on, it's about 2.5 centimeter resolution. Here's what hadal sediment cores look like. Rapid depletion of oxygen. So that's the oxic zone, and then eventually sequential rapid depletion of nitrate due to nitrate reduction. And then we then start to see manganese, and then ammonia, and then iron, indicating this ferruginous zone. And this actually very nicely reflects what sediments where oxygen is rapidly turned over in the surface sediments looks like. So there is this nice transition from oxic zone where organisms are mostly using oxygen to respire. And then there's a transition into this nitrogenous, then manganese and ferruginous zone where other metabolisms become more prevalent. So nitrate reduction, manganese reduction, as well as iron reduction. They don't quite observe the sulfatic zone as well as the methanic zone. But nevertheless, what this results in is very steep changes in microbial community structure. So this is an this is a PCOA plot. And I believe they use Bray-Curtis dissimilarity here. And the colors indicate sediment depth such that darker colors are shallower sediments, or sorry, the surface of this sediment, and then going deeper into the sediment transitions into the yellow. And then this also incorporates all of the different sites that they looked at the Atacama Trench. So essentially what this shows us, when just looking at one trench, is that depth differences are, one trench, but multiple sites within the trench, is that depth differences are greater than site differences in this one trench. But when they compared this microbial community to other trenches, so Kermitic Trench, as well as other realms, so the bathyal, as well as abyssal, you can see a very nice separation of the bathyal and the abyssal, which looks very similar to each other. And then here is the Hadal environment in yellow, and there's even a nice separation between the Kermitic Trench microbial community from the Atacama Trench microbial community, indicating quite a bit of heterogeneity even across trenches. This is a paper that just came out, which unfortunately is beyond the scope of this talk, but I highly encourage you all to I highly encourage you all to look into this because they've got metagenomes, they've got bacterial and fungal isolates, and they did quite a bit of viral work as well. Here are just sort of some representative figures here, but it's a really nice and comprehensive characterization of the biosphere in the Challenger Deep, which is that deepest part in the Mariana Trench. Essentially just a few highlights. They show very versatile lifestyles based on metagenome assembled genomes, versatile lifestyles of mixotrophy, as well as facultative anaerobic metabolism. And that's because they need to be able to rely on these anaerobic metabolisms due to the rapid depletion of oxygen. They also show some fungal groups that have been identified for the first time in Hadal environments. And then they also show a range of viral auxiliary genes that essentially suggest that these viruses are potentially making use of the host's nitrogen and sulfur metabolism. It's a really interesting paper, and I encourage you all to look into it. And then they have a range of isolates that anyone, I'm sure, can play around with. So that is a culmination. That is the end of the introductory part. And hopefully that gives us a little bit of time. Do we have a little bit of time? I think, oh no, that was already 30 minutes. Oh no.
I think you could keep going. Sorry.
Okay. I think it will take about 10 minutes if that's okay.
That's good.
Perfect. Good. So the brief recap of Hadal environments, and then we'll transition into a brief talk about my current research. Essentially, due to their bathymetry or the steep slopes, Hadal trenches act as funnels for material deposition. There's elevated carbon deposition from sinking particles, movement laterally, as well as mass wasting events that lead to high carbon accumulation rates in Hadal sediments. There are a lot of unique abiotic parameters in trenches that lead to very different kinds of pelagic and benthic microbial communities, and that these benthic microbial communities and their metabolic activities are highly influenced by the sharp redox gradients, in large part due to the rapid depletion of oxygen. Okay, so based on this recap, one thing that is actually kind of a glaringly obvious question is, if this sinking organic matter is easily consumed, right? If it reaches this the deep sea, and is very highly consumed, easily consumed, or highly labile, why does it persist during descent? Okay, so that's a crux. That's the core question of the talk here. And one hypothesis that we have is that high hydrostatic pressure acts so that it limits or reduces microbial activities, particularly the activities of surface originating microorganisms, such as those that are sinking attached to particles, or those that are displaced from shallower sediments down to the Hadal sediments due to mass wasting events. So the microbial activity that I'm interested in here is the enzymatic degradation of organic matter. It is the first step of the degradation of organic matter, so it's necessary. Here is sort of a schematic of a bacterium. Let me just switch back to laser pointer. Here's a schematic of a bacterium kind of encountering its food in the form of particulate organic matter, sort of thinking, how in the world will I get this to go into my cell? Right? Because it's massive. And the way that they can do this is actually by producing their own utensils, which we would call these hydrolytic enzymes, represented here in the black, that break down these large chunks of organic matter into much smaller chunks called dissolved organic matter. And once it's in these dissolved form and much smaller, they can then assimilate that into their cells or use it to respire. And the questions that we are interested in, if this thing will let me, here you go. The questions that we're interested in are relevant to microorganisms that are descending, right, from the surface of the water or from shallower sediments. So the first question that we want to ask is, does increasing pressure inhibit, stimulate, or have no effect on microbial hydrolytic enzymes, which are necessary for organic matter degradation? How do increasing pressures affect the enzymatic activities of complex pelagic and benthic microbial communities? And to what extent are pressure effects related to the production of enzymes? Does it slow down the production of enzymes by microorganisms, or does it just affect the catalytic efficiency of enzymes once these enzymes are produced? Essentially, does it act on the physical structure of the enzymes, therefore reducing its activity? I want to highlight that this builds on quite a bit of prior work, and I highly encourage you all to look into these papers and book chapters. I think they do a phenomenal job in bringing us up to date with what's been done. So how do we test pressure effects on microbial hydrolytic enzymes? First, I want to highlight that we measure quite a number of different kinds of enzymes. And essentially what we do is we take a substrate that we know measures the activities of these specific enzymes, and we add it to our sample, be it water, seawater, be it sediments, and then we do a range of measurements, either based on fluorescence, changes in fluorescence, as well as changes in molecular weight. We target these kinds of enzymes because we know that marine organic matter is composed of these different kinds of constituents, and therefore these enzymes are likely to be active in the marine environment and relevant for the marine environment. And what we do with these assays, with these incubations, with our sample, with a substrate added to them, is we then put these samples into these pressure vessels. And we run these incubations for about 24 hours in the cold, in the dark. And we place them in multiple pressure vessels so that we can mimic the different levels of pressure that sinking microorganisms encounter in the ocean, going all the way from a control of atmospheric, so no increase in pressure, all the way down to 100 megapascals, which is the pressure that microorganisms experience at 10,000 meters of water. And just for a brief look at what these instruments look like, here's a pressure chamber with with the capacity to measure oxygen measurement. So this is actually really nice for wanting to measure oxygen consumption while things are pressurized. The one at the bottom is a pressure cell, and this one can measure fluorescence while things are pressurized. And this one is just a fairly simple but fairly robust pressure cylinder that was designed at Scripps in San Diego. So these are just some of the pressure vessels that we use in this study. So delving right into the results, what pressure-related responses might we expect? So HHP indicates high hydrostatic pressure. So it's some increased pressure that's above what ATM is, or atmospheric pressure. And then we take the ratio of the activity at these different high hydrostatic pressure levels, divide it by or relate it to the atmospheric pressure, multiply it, the rate at the atmospheric pressure, and then we multiply it by 100%. So that if we see that this is less than 100%, we see some pressure-related inhibition. If this ratio is the same or 100%, it indicates pressure-related insensitivity. The activity simply doesn't care about the pressure, or it can withstand the pressure. And then this one is great. If it's greater than 100%, then maybe there might be some pressure-related stimulation in activity, which is kind of a little bit difficult to wrap one's head around. But essentially, in other words, this is percent activity at high hydrostatic pressure relative to activity at atmospheric pressure, which is at 0.1 megapascals. So we first do this by looking at a pure culture called Photobacterium profundum. And so this is a proof of concept where we grew these. This is the information from Photobacterium profundum. This is some details about it. It comes from the Seleucia at 2,500 meters. It's a moderate piezophile, so it grows optimally at 28 megapascals, so 2,800 meters equivalent of pressure. And so I measured activities of these different enzymes: beta-glucosidase, leucine-aminopeptidase, NAG for chitinase, and phosphatase. And this is the different pressure levels at which I measured activity. And so you can see that leucine-aminopeptidase, chitinase, as well as phosphatase become fairly stimulated at its optimal growth pressure at 28 megapascals. So you get very high rates of activity. And then eventually, the more that you subject it to increasing pressure, the lower its activity becomes. And so when you take that ratio of activity at some high hydrostatic pressure and relate it back to its activity at atmospheric pressure, this is what you get. 100% or insensitivity is indicated by this red dashed line. And so you see that 28 megapascals and 50 megapascals, you still see stimulation. But once you subject it to 75 and 100 megapascals, you actually see very strong inhibition, even of this moderate piezophile. So essentially, this one organism can exhibit a wide range of responses. You can see the stimulation at 28 and 50 megapascal, inhibition at 75 and 100 megapascals, and you get very different degrees of stimulation. So you can have about 424% stimulation, and then substantial inhibition by leucine-aminopeptidase, or you can get fairly subtle stimulation by phosphatase at 171%. And then with chitinase, you see 204% stimulation, but then not so much inhibition. It's still able to maintain 53% of its activity, which to me is just really, really interesting that the same piezophile can have such dramatic responses depending on what pressure you subject it to. So how does this pressure effect look like for complex pelagic and benthic microbial communities? Well, we did these experiments at a range of places between last year and this year. And this is the compilation of that. And so it's the same plot, but with the same enzymes, beta-glucosidase, leucine-aminopeptidase, and chitinase, except what you're seeing here is the seawater in the surface, or sorry, at the top over here, and then sediment in the bottom. And the things that I want to highlight is that the effects are very variable, but actually the most common response is some level of pressure-related inhibition. So these dots lie to the left of the red line. But actually what we see is that there are some surface originating microorganisms, especially among sediments, that can maintain function at Hadal environments. So some of these complex sediment communities are actually also showing activities, stimulated activities at high pressure, despite them coming from shallow coastal sediments. And what this indicates to me is that there might be potentially some underestimated piezo-tolerant trait among complex microbial assemblages in surface sediments. which is really intriguing because even that moderate piezophile that we looked at can be severely inhibited at pressures of greater than 50 megapascals. But some of these shallow sediment microbial communities are actually stimulated at 75 megapascals. So they're doing better than this moderate piezophile. Now, to this third question, to what extent are pressure effects related to the production of enzymes or the impact of pressure on enzyme efficiency? We went about this in a sort of indirect way, where we stimulated enzyme production using two different particulate organic matter sources, a plant biomass and a diatom biomass. And then we separated those cells from their the enzymes from their producing cells. We stimulated enzyme production and we separated those enzymes from their producing cells. So we remove the cells and we only measure the pure dissolved enzyme activity in response to pressure. And here are the results for that. So beta-glucosidase activities, leucine aminopeptidase, and chitinase. And what we see very nicely is mostly a decrease. We see some tentative outlier here. So it looks to me that these are all inhibition, right? So consistent pressure-related reduction or inhibition, perhaps with some tentative outliers, suggests to me that pressure may actually act more on enzymes and reducing their catalytic efficiency rather than on the production of enzymes. And so I thought my life was over, this study is complete, so on and so forth. And then I looked at the data for dissolved enzymes produced by the marine POM. And what we see is actually, there's mostly inhibition, but there's actually some level of pressure tolerance here as well. And we're measuring the same enzyme activity here, leucine aminopeptidase and chitinase, except when it's stimulated by by terrestrial POM, it's highly pressure sensitive, but when it's produced and stimulated by marine POM, it's actually relatively pressure resistant. So that's what that's saying. And essentially, I think the final or the conclusion here for this slide is that enzymes have very different pressure sensitivities. And actually, we think that it's a function of the pressure tolerance or the sensitivity of their producing taxa. And that's how we can have such different responses for the same enzyme activity. It really depends on which organism produced them. Now, I want to take a step back from just enzyme activities and look at another step in organic matter remineralization, which is that final respiration step. So I've only shown you that initial enzymatic degradation, but I also want to compare it to pressure effects on the respiration, oxygen respiration. So here is the compilation of all of my results that I just shared with you. And here is a similar study design, but measuring respiration, conducted by Peter Steve here at University of Southern Denmark. And what he sees is that this relative respiration, that's the same ratio at some high hydrostatic pressure divided by the rate at atmospheric pressure, is out in seawater. Once you reach about 7,000 meters or 70 megapascals, down to 100 megapascals, you're essentially seeing like near or complete inhibition of respiration. And this is a little bit deeper for microorganisms that are associated with aggregates. You see near complete or complete inhibition of respiration at 90 or 100 megapascals, which when we compare that to the enzyme activity, we notice that at high pressures, it looks like enzymatic degradation of organic matter by surface-associated microorganisms may actually still proceed, albeit at reduced levels, but respiration may be halted. So these enzymes can continue breaking down the organic matter, but the organisms cannot then continue to respire it and bring it back to carbon dioxide, I think is what this is saying. So to end this talk, just to summarize, here was our hypothesis, thinking that hydrostatic pressure just limits microbial activities. And we can only partially accept this. We see that the most frequent effect of pressure is some level of activity reduction, which kind of goes back to this idea of helping preserve organic matter during descent. So we kind of partially accept that, but actually the devil is in the details. When we look at specific questions, we see that yes, we can see pressure inhibition, pressure stimulation, or pressure insensitivity. And we think that this pressure effect varies as a function of which enzymes we're looking at, as well as the pressure sensitivity of the microorganisms that are producing the enzymes. How do the pressure effects compare for water column versus sediment microbial communities? It seems like they're similar, but the activity reduction is greater for pelagic microbial communities. And some members of benthic microbial communities, probably due to their high diversity, may have an underestimated potential for pressure tolerance, which to me is fairly new. And to what extent are pressure effects related to the production of enzymes or the impact of pressure on enzyme efficiency? To a limited extent for both. We see evidence of pressure sensitivity and insensitivity for freely released enzymes. But one key takeaway is that even if their activity is reduced, some of these enzymes that are produced in surface waters and decoupled from their producers may still be able to function at Hadal depths. So that to me is really surprising and mind-boggling. So that ends my really long talk. Thank you so much to all these folks. This is an outdated version of the members of the Hadal Center here at the University of Southern Denmark. Some of my collaborators, Carol Arnasi, Stephanie, and Doug Bartlett. Thank you so much, Sari, for the invitation. And I want to thank these institutions and these funding agencies for their support in this project. And thanks to all of you for listening.